Hi, welcome back to Chapter 11. This week we'll be starting our look at the topic of genetics, and in this chapter we are covering the first geneticist, Mendel, and going through his discoveries. First up is Mendel and the Black Box, 11.1. And Gregor Mendel, which is this man here, is considered the father of genetics and was the first person to comprehend some of the most basic principles of genetics. When we talk about genetics, we are talking about how traits are passed down from one generation to the next. And they knew that traits were passed down from one generation to the next at that point. They just did not understand how that worked. And Mendel was born in 1822 in Heisendorf, Austria, and his name at birth was Johann Mendel, and he was born into a poor farming family. At that time, it was difficult for poor families to obtain a good education, so Mendel saw the only way to escape the life of poverty was to enter the monastery. So he entered the monastery at St. Thomas's Abbey in Brno in what is now Czechoslovakia, and here he was given the name Gregor. The monastery was in the Augustinian Order of St. Thomas, which was a teaching order and had a reputation as a center of learning and scientific inquiry. This is a picture of the abbey here that he went to at that time. And to enable him to further his education, the abbot of the abbey arranged for Mendel to attend the University of Vienna. And Mendel was nervous here. He did not perform well. Remember, he was from a very poor background, and most of the people going to a big university at that point were from very wealthy families. So he really did not fit in that sense, but that did make him nervous and not do very well because of that. His examiner there at the university failed him and said that he lacked insight. And so in 1853, he had to return to the monastery as a failure. And since this was a teaching order, that was sort of the goal, was he would get an education and teach. And so Mendel had to decide whether he would stay on at the monastery as a failed teacher or go back to the monastery and do what there. He really didn't know at that point. But one thing that did help shape his path while he was at the university was a doctor and professor there at the university named Franz Unger, and he was teaching botany courses at that point at the university, and Mendel learned about his practical view of inheritance, which, free from spiritual influences of what they thought was going on at the time, this practical view of inheritance seemed to reflect his own farming background, so things that he had seen of how traits get passed along. So this gave Mendel the idea to stay on at the monastery and use his time to carry out practical experiments in biology. Okay, so next up is section 11.2, the experimental subjects, Pisum sativum, which is the common pea plant. After about 2 years of being back at the monastery, Mendel began his investigation into variation, heredity, and evolution in plants. He chose to study in detail the common garden pea, Pisum, which he grew in the monastery garden. And this is a picture on the right of the garden at the monastery where he worked. Between 1856 and 1863, Mendel patiently cultivated and tested at least 20,000 pea plants, analyzing the seeds in the plants and comparing traits for things such as seed shape, seed color, whether they were tall or short-stemmed plants, the color of the flowers, and so 11.3 goes into starting his experiments there. And the main thing that he had to do when he started his experiments was breed true plants. So he wanted plants that were bred true. And what this meant was that those plants only have just the trait he's looking for. So for example, if he was wanting to study purple flowered pea plants, he would grow those plants through many generations and make sure that every generation of purple parents, so if you cross two purple flower pea plants, that all the offspring was purple. So you weren't occasionally getting a white flower or another color flower coming out. So bred true means that you only get that one trait that you're looking for. What we now know is that those plants have 2 copies of the purple flower gene. So that's why they're always purple. At the time, he didn't know that's what was going on. He just knew that You could get these, what he called bred true plants, if you made sure that that generation after generation came out the same as the parents. And again, that's what he was looking for in his studies. So to cross-pollinate the plants, Mendel worked for several years carefully cross-pollinating. So he didn't just let the wind blow pollen from one plant to another, because he wanted to know that exactly the plants that he wanted to cross were the plants that were actually being crossed. So he would take, let's say he was looking for the trait of yellow seeds and wanted to cross it with a green seed. So he would take the pollen very carefully and peel back the closed petals of the pea plant and pull off some of the pollen with some tweezers, and then take the flower grown from the green seed and paint that pollen on. So naturally this would happen by insects out in the outside, or by the wind, but he did this with tweezers and a paintbrush to make sure that he crossed the two exact plants he was wanting to cross. So the results of this is that he was making sure, or the goal of this is to make sure that he was crossing the two plants that he actually wanted to cross. Okay, so when he crossed plants, he started noticing some trends. And in this example, we're going to look at his cross of tall and short plants. And he noticed that if he crossed bred true tall plants, so plants that came from all tall generations, with short plants, so if you had one parent that was tall, a tall pea plant, and one parent that was short pea plant, that all of the next generation came out tall. They call the parent generation the P for parent generation, and they call the offspring the F generation. So F1 would mean the first generation of offspring from these parents. And so what was important here is that he noticed that all the offspring came out tall, rather than being a medium height blend, which you might expect from just mixing the two plants together. So this first generation, he's noticing all came out tall when you mixed a tall and short parent. Now he found that when he crossed the offspring from those plants, so if you were to take some of these all tall offsprings from this F1 generation and cross two of those plants together, that in the next generation, the F2, or the second generation of offspring, that he always got the ratio of 3 tall plants to 1 short plant. And again, the traits didn't blend. He didn't end up with medium plants, but he noticed this pattern where the first generation, when you mix plants with two different traits, the first generation all came out tall, and that second generation, when you mix two of these tall plants, came out 3 tall and 1 short. Mendel's observation led him to coin 2 terms that we still use in present-day genetics, and he coined the terms dominant and recessive for traits. And he realized that with each character he studied, there are 2 varieties or traits, and one of them is dominant, meaning it shows up even when the other trait is present, and the other trait is recessive, and that trait does not, he noticed, did not show up right away. So when 2 traits were crossed, he noticed the dominant trait would show up in the first generation. So he's looking at things such as flower color, mixing purple and white flowers, and he's noticing if you mix purple and white, that all that next generation is going to come out purple. And when he mixed tall and short plants, all of that first generation came out tall. And so with every trait that he noticed, there was one, with whether he's looking at seed color or seed shape or plant height, that there was one trait that always, whatever that trait was that showed up in that first generation, he named that the dominant trait. Now, again, the dominant trait shows up in the first generation, and so in our tall and short plants, the tall plants would be considered dominant because they show up in the first generation. And the short plants were recessive, and they did not show up in this first generation. Now, it would have been easy to think that that short trait just isn't there at all in that plant, but he noticed that when you get to the next generation, again, and you're mixing 2 of these tall parent plants, that they don't come out all tall, which is what would happen when you would cross just bred true tall plants. He noticed that you get 3 tall plants, and so 3 plants with the dominant trait and 1 plant with the recessive trait. And he saw this pattern over and over again in all the traits that he looked at. So for example, if you're mixing purple and white flowers, the first generation would have all purple flowers, and so all of that dominant trait. The next generation would have 3 plants with purple flowers and 1 plant with white flowers. So again, he coined these terms dominant and recessive, and dominant being the trait that shows up more of the time and shows up especially specifically in this first generation, and recessive being the trait that's there but not showing up all the time. One of Mendel's central insights was that the basic units of genetics are material elements. So we now know that we have DNA. This DNA is made up of pieces called genes. He didn't know that at the time, but he did know that there was something in there and that it was in pairs. We know that as being pairs of genes on chromosomes. So you have 2 pairs of chromosomes, one from mom, one from dad. Again, he did not know that at that point. He just knew that there was something in there and that there was a pair of them, and one came from one of the parents and one came from the other parent. Today, we call these elements genes, and they come in alternative forms called allele. So you might have an allele for a tall gene and an allele for a short gene. So again, allele just means the form of the gene that you're getting, that specific trait. Mendel noticed that plants had a physical appearance, what we now call the phenotype, and this differed from something inside the plants, the alleles which make up what we now call the genotype, that was being passed down to determine the traits. So we have the terms that we use, and this is what Mendel noticed, that we see here tall and short. We see here in this generation all tall. So phenotype is just what you see on the outside. So this plant being tall, that's its phenotype. This plant has the same phenotype as this plant. They're both tall plants. What's different about this plant and this plant is that they have a different genotype. So this plant has all tall genes in it. This plant has a tall gene from one parent and a short gene from another parent. So its genotype is to have a tall gene and a short gene. So again, phenotype tall, also phenotype tall. The genotype, though, is different. So let's look at how we identify and write down that genotype next. Again, each trait is made up from 2 genes, one from mom and one from dad. Pure tall plants have 2 tall genes. We label the dominant trait with a capital letter. So remember, tall was our dominant trait. So tall plants, we would say they have 2 pure tall plants, bred pure. We would say have 2 tall genes, or capital T, capital T. Again, we write the letter as a capital if it's the dominant gene. Pure short plants have 2 short genes. We label the recessive trait with a lowercase letter. So short plants, we would say, have the genes bred pure short plants, lowercase t, lowercase d. So again, big T for the dominant bred pure plant, and little t for the recessive short bred pure plant. So let's look at what's going on with, so we've looked at the phenotype in the different generations, so whether the plants are tall or short. Let's look next at what the genes are in each of those generations. So the tall plant with genes big T, will undergo meiosis, and again, meiosis is when that egg of basically your seed, which is like an egg in humans, will undergo meiosis and produce seeds or pollen, each with a T gene. So this tall plant has 2 T genes. When this splits apart to make the seed or the pollen, each of those T genes will go into a different cell to get ready to be passed on and mix with the other plant that it gets fertilized by. So The short plant with genes lowercase t will also undergo meiosis, producing seeds or pollen that each have a little t gene. So when you cross a tall plant, let's say seed, with a short plant pollen, so a big T with a little t, then all of the plants that come from that are going to get a big T gene from one parent and a little t gene from the other parent. So all of the offspring therefore have big T, little t, and remember the tall trait is dominant, so that's the trait that we see in the phenotype, or what we're seeing, what the organism looks like. So this is why the first generation all comes out tall, because they all will have big T, little t as their genes. A tall and a short gene, tall is dominant, tall shows up. So genotype is big T, little t, phenotype is tall. So remember, all the offspring from that first cross have the genotype big T, little t. So when we cross two parents who are both big T, little t, so again, they're both the two tall parents. Let's see what happens with their offspring. So eggs or pollen will, from here, will either have a big T or a little t. So this cell will undergo meiosis. The big T will go into one cell. The little t will go into the other cell. And also this parent will undergo meiosis, and the same thing will happen. So you'll have a big T and you'll have a little t. So when you cross these plants, again, the eggs or pollen have big T or little t. So the possible offspring that result from this mix, so this parent could give a big T or a little t to an offspring, and this parent could give a big T or a little t for an offspring. So the possible combinations in the offspring that you get when you're just looking at this gene are that the offspring could get a big T from each parent. They would have the genotype big T, and the phenotype tall. You could also get the offspring getting a little t from this parent and a big T from this parent. They would have the offspring big T, little t, and the phenotype also tall. You could also have the offspring getting a big T from this parent or a little t from this parent. They would have the genotype again, big T, little t, and the phenotype tall. And our last possible combination from these two parents is that the offspring could get a little t from this parent, a little t from this parent. They'd have the genotype little t, and they would be short. So one of these offspring out of every four will be short. Two of the offspring will have big T, little t, and be tall. And the third offspring will have big T, and be tall. So 3 tall offspring and 1 short offspring. Let's review that. Our parents, our P generation, had tall and short, big T for the tall, little t for the short. So all of their offspring got one of these big T's from mom, one of these little t's from dad, and all came out big T, little t, which is tall. And all of our second generation of offspring came out, sorry, not all of them, but 3 of the second generation came out tall, 1 short, and again, for that second generation, we have 1 plant that's big T, 2 plants that are big T, little t, and 1 plant that is little t. Next up is section 11.4, looking at the next generation. And let's try crossing a parent. So doing some different crosses with genes and seeing what happens. So if you have a parent with big T, little t, so a tall parent, but not bred true tall, and a short parent, little t, let's see what happens. And we're going to do this cross instead of drawing all the lines like we did with the other example and going through meiosis. We're going to do something called a Punnett square. That's this square here, looks sort of like a window pane. And it helps us to organize our genes from the parents and see what the genes from the offspring will look like. So what we're going to do is take the genes from one parent, and place them on one side, and it does not matter what side you place them on. So let's take our first parent with big T, little t, and we're going to place one gene on the top row, and the next gene on the bottom row. So we write them right here on the side, and these are the parent's genes from one of the parents. The next thing that we do is take the genes from the other parent. So if the other parent is little t, we take those little t's and we put them on the top of the Punnett square. So we put a little t here and a little t here. And again, you could have put big T, little t up here and little t on this side. It would not have mattered as long as one parent's genes go on one side and one parent's genes go on the top. Now we're going to move our genes from the top and the side and move them into the Punnett square to see what the genes of our offspring. All right, we've got our parents' genes on either side of the Punnett square, and now we're going to move these genes on the top and the side. We're going to move them into the Punnett square. And so the way this works is for the first upper left-hand corner box, we're going to take the trait from the top and we're going to move it down. So the traits, these little t's are going to get moved down into each of these boxes. And on the side, this big T will get moved over into this box, and it will also get moved over into this box. So to fill out this first box, we move our big T over, we move our little t down, and we get a big T, little t in this box. Now for the next box, down here, the lower left-hand box, we move this little t down and this little t over. For this upper right-hand box, we move the little t down and we move the big T over and we get big T, little t in that box. And finally, for our bottom right-hand corner, we move the little t down and the little t over and get little t in that box. Now in our Punnett square, each of these boxes here shows the proportion of offspring that will have a certain genotype. So it shows all the different genotype possibilities for all of our offspring. So if you mix little t short plant with big T little t tall plant, you will get these 4 different types of offspring. You'll get some of the offspring will have big T little t, some others of the offspring will have big T little t, and then offspring will have little t, and other offspring will have little t. And what these boxes show us are the percentage of offspring that will have each of these traits. So we see that 2 out of our 4 total offspring, so we have 4 total possible combinations of offspring, and 2 of the 4 will have big T, little t. Now 2 out of 4, if you can do some math here, is half, or 50%. So 50% of our offspring will come out with the genotype big T, little t. 2 out of 4 of our offspring, so 2 out of the 4 boxes, remember each box is an offspring, 2 out of the 4 boxes will have little t, so we'll have 50% of our offspring in this cross. Again, these 2 parents will be little t will be short. So the genotype is 50% big T, little t, and 50% little t. The phenotype is 50% tall, because all of these big T, little t's will be tall, and 50% short, because all of the little t's will be short. Now, Punnett squares are something that just takes some practice with, getting used to putting the traits on the side and the top, moving them down, and then analyzing what the offspring will look like. So I will have some practice problems posted this week for you to work on, and you will see these on your next test, so please make sure that you practiced until you are comfortable doing them. I'm going to go through a couple of the things that Mendel realized as he did his studies, and again, one that we discussed is that genes retain their character through many generations rather than being blended together. So these, for example, tall and short genes get passed on. They don't just get blended together. The tall genes get passed on as a specific entity, and the short genes get passed on as a specific entity. He also noticed something called the law of segregation, which is that alleles separate prior to the formation of gametes. We now know that this is in meiosis, that those 2 genes that each parent has separate into 2 different cells, whether that's the egg and the sperm, or the egg and the seed, or the seed and the pollen, and these genes get separated prior to that forming of the gamete, the egg or the seed and the pollen in plants. So the alleles Mendel was observing um, reside on, as we now know, homologous chromosomes. So that's chromosomes that are similar, but again, you have one from mom and one from dad, and these separated in meiosis. This concept is known as the law of segregation, where the two traits separate and you get one trait in one of the gametes and one trait in another one of the. An organism that has two identical alleles of a gene for a given character is said to be homozygous for that character. So for example, if you have two tall genes or two short genes, then you'd be homozygous tall or homozygous short. An organism that has differing alleles for a character is said to be heterozygous for that character. So that would mean, in our example of tall and short, that you have one tall gene and one short gene. So again, homozygous means you have two of the same trait. Your two genes are both the same trait, and heterozygous is the two gene copies that you have are from two different traits. The dominant trait is expressed in the heterozygous condition. So what this is saying is that when you have, for example, big T and little t, so a tall and a short gene, what you're going to see is the dominant trait. So you will see that plant as being tall. Now, recessive is not expressed in the heterozygous condition. So when you have a big T, little t, a short and tall plant gene within a plant, you do not see the recessive trait. So for example, the short P's are absent in the big T, little t condition. So when you have big T, little t gene, you do not see the recessive trait. So again, heterozygous means you have two different copies of a gene, and when you have two different copies of a gene, you're going to see the dominant trait and not see the recessive trait. Let's look at a little bit more detail on alleles. And one allele, copy of a gene that gives a certain trait, resides on one chromosome. So if we're looking at our chromosome here, and we have one chromosome from mom and one chromosome from dad, one of the alleles resides on one chromosome, and the other allele resides on a similar place on the other chromosome. So in this example here, if you have an allele for a yellow seeded plant from mom and an allele for a yellow seeded plant from dad, this combination of alleles would be considered homozygous dominant. Again, homozygous means the same trait on each chromosome, and the trait that it has is the dominant trait. So yellow is our dominant trait, and that's in this example here. Now, if you were to have a chromosome from mom that has the yellow allele and a chromosome from dad that has the green allele, then you would be considered heterozygous because you have two different alleles for that trait. And again, the dominant trait will show up in that plant. So that plant would have yellow seeds because yellow is the dominant trait. Now in our last example here, we have this plant having one copy of a green seeded allele from mom, copy of a green seeded allele from dad, and this would also be considered homozygous. So this was homozygous. This is heterozygous. This is homozygous again because you have two of the same trait. So you have two green alleles, you are considered homozygous, but instead of being homozygous dominant like this plant, it's homozygous recessive because this is the recessive trait. And the recessive trait is for green seeds, so in this condition, green seeds show up. So again, just keep these terms in mind. Homozygous dominant, two of the same dominant trait. Homozygous recessive, two of the same recessive trait. And heterozygous means you have one that you have two different traits. Okay, so again, just to sum up, homozygous dominant means you have two dominant alleles. Homozygous recessive means you have two recessive alleles. And when you have two different alleles, you are called heterozygous. Next, let's look at section 11.5, crosses involving two characters. So we have looked at one character only being studied in the plant. For example, whether the plant is tall or short, but plants have multiple characters, and all of these characters are going to separate in meiosis and get carried down from one generation to the next. Now, Mendel noticed something else, that when you're looking at plants with multiple characters, that the genes for the different characters he studied were passed on independently of one another. So, for example, when you mix a plant with yellow seeds that are also round and green seeds that are wrinkled, that offspring could have any combination of yellow and round, or green and wrinkled genes. So the plants didn't always stay yellow and round. They could end up throughout the generation as yellow and wrinkled. And green plants didn't always stay wrinkled. You could get green and wrinkled plants, or you could get green and round plants. And the reason that these traits don't always stay together, so for example, you don't always get yellow round seeds, is that the characters for these traits reside on separate non-homologous chromosomes. So on one chromosome, there is, for example, the round allele, and another different chromosome would have the yellow allele. So when these plants undergo meiosis and these genes separate out, the seeds or pollen could end up with either one of these genes being passed on. And again, that traits can mix, that the traits don't always stay together. So for example, in humans, that might mean that you don't always have to stay brown-haired and brown-eyed, or blonde hair and blue-eyed. These traits can mix and you can end up with blonde hair and brown eyes, or brown hair and blue eyes. And this is because of something called independent assortment. So an independent assortment means that the traits are located on different chromosomes, and so those different chromosomes can get passed down in any combination to the gametes and to the offspring. So Mendel had all of these great ideas, things that were completely new to his time. People had never thought of these ideas, and section 11.6 goes into how his ideas were received by the scientific community. And in 1866, he published his work, and it went completely unnoticed. This was too new and complex of an idea, and even influential scientists could not relate to this idea. So again, people at this time, you couldn't see DNA, you couldn't see genes. They had no idea what was going on inside the plant. He just knew from looking at what he studied that this must be what's going on. But people really, even educated scientists, could not relate to this. Mendel from this point went on to run the monastery. Again, this was a pretty controversial subject at the time, looking at biology and traits. And when Mendel died, the new abbot of the monastery burned all of his papers. And so you would think that might have been the end of Mendel. Luckily, 16 years after he died in the year 1900, his work was recognized by 3 different Dutch botanists. And it still, though, wasn't until the 1920s and 30s that the importance of his work really was understood, and that was that it was a foundation for explaining the Darwinian theory of evolution. And that's when they realized that you need genes to explain how traits get passed on and how populations can change. And we'll talk more on that in future chapters in the text, but again, it took a long time for this topic to be really understood and realize how significant it is. Now we consider him the father of genetics. We realize that this is the basis for all of our genetic studying. It's a hugely important topic, but again, at the time, they didn't have the background to understand that, and did not realize how important that his work would later be. Now, section 11.7 goes into incomplete dominance and codominance. So Mendel studied pea plants that worked very nicely because the traits got passed on as either the dominant trait showed up in the first generation, recessive trait didn't show up in the first generation. But if you look at other plants, you can get blending of genes, which he did not get in the pea plants. So in some genes, neither allele is completely dominant, and that will mean that the heterozygous genotype, so again, when they have one trait for mom and a different trait for dad, they can be intermediate in phenotype, so they'll look like a blend of the parents. So in the snapdragon plant, a red parent, big R big R and a white parent, little r, will end up with some offspring who have big R, little r will be pink. They'll be a blend. So the big R offspring will still be red. The little r offspring will be white, but the big R, little r will have a blend and be pink. And this is called incomplete dominance. So the red gene is not completely dominant like we would have seen in the pea plant. Now, some genes you can also get something called codominance, and this is when different alleles in the gene will have independent effects on the organism. This is the case in our gene that codes for our blood type. So for whether you're type A, B, or O, and these are these glycolipids that are on the surface of your blood cell. So if you are if you have the two genes AA or AO, you will have these A glycolipids on all of your blood cells. If you are BB or BO, then you will have the B glycolipids on your blood cells. If you are blood type AB, you will have both A and B glycolipids. And if you are OO, you will have no surface glycolipids. And neither allele is dominant, but each allele does something different and makes your blood look a little bit different. So both traits are equally present in codominance. Section 11.8 looks at multiple alleles and polygenic inheritance. Human beings and many other species can have no more than 2 alleles for a given gene, and each allele resides on a separate homologous chromosome. For example, you have a trait, you have one chromosome from mom, one chromosome from dad. Each of those have a trait on them, but there can be you can have many different types of genes in the population. So for example, if you're looking at eye color, you can have green and blue and brown and any combination in between there and mix of eye colors. So you can have lots of different eye colors, but for any given gene, you only carry 2 copies of that trait, one from mom and one from dad. But most traits in living things are governed by many genes. So we looked at in a previous chapter eye color, and eye color is controlled by a whole wide array of genes, and this is known as polygenic inheritance, and that means that the inheritance of a genetic character is determined by the interaction of multiple genes, each having a small additive effect on the character. So if you're looking at eye color, you might be looking at 20, 30 genes, and those all produce something different to add to that overall trait of your eye color. And again, that's polygenic inheritance. Your height is the same way. Your height, you don't just have a tall and a short gene in humans. There's a whole bunch of different factors going into the genes that produce your overall height. Now, polygenic inheritance tends to produce continuous variation of phenotypes in which there are no fixed increments of differences between individuals. And what this means is that in the pea plant, we saw tall and short plants. But in most organisms, when you're looking at a certain trait, you're going to see a whole range everywhere from if you were looking at, say, height, very, very short to very, very tall, and everything in between. So human skin, for example, comes in a range of colors in which no color, one color shades imperceptibly to the next. So you could have every mix of possible skin colors, not just light or dark. Now, the traits produced in polygenic inheritance tend to manifest in bell curve distributions, in which most individuals display near the average trait rather than at the extreme. So for example, if you are looking at human height, you will have a few people who are pretty on the short end of the height, average height, and a few people who are on the very tall end of height. But most of the people will be of this average height for humans. And this, it looks like if you were to look at a population in general, you have a few short, getting more and more people as you go toward the medium, and then fewer and fewer as you go toward the tall. This makes sort of the shape of a bell. And this would be the shape here that we're looking at. When you graph the number of individuals of each height, you get this bell shape. The same thing happens if you were to say, look at beak depth of a certain type of bird. Some birds will have very narrow beaks, some birds will have very wide beaks, but most birds will have sort of this average beak. And this bell curve that we see here tends to happen when a trait is due to polygenic inheritance, meaning a trait is governed by or controlled by multiple genes. If a trait is just controlled by one gene, such as the pea plant that Mendel looked at, then you'll just see all tall and all short, and nothing in between. But the bell shape is from multiple genes determining that trait. Gene interactions and gene-environment interactions are so complex in polygenic inheritance that predictions about phenotypes are really we can't predict what a phenotype is going to be. So you can't look at two parents and know for sure what all their offspring will look like. But we can say that they will have a certain probability of looking a certain way. So if you were to look at the genes of the two parents and their traits, you could say the children will probably come out with these certain characteristics, but it's a probability and not a certainty because there are so many gene possibilities involved. Now, section 11.9 looks at how environment affects the genes. And so we have certain genes that we have on our body and plants have in their bodies, but the environment can sometimes impact how those genes. So for example, in the hydrangeas here, the acidity of the soil in which it lives in can determine whether they are pink or blue. For example, if you have a more acidic soil, a pH 5.5 or lower, you'll get blue flowered hydrangeas. If you have a pH of 6.5 or higher, so an alkaline or basic soil, you will have pink flowers. And if you want a mix of purple and sort of purple blooms, then you would need the pH to be between 5.5 and 6.5. So that's just one example of how a trait is not just determined by the genes that it has, but traits can also be determined by the environment. That is the end of our chapter 11 lecture. So please let me know if you need any help as you go on about your week, and we will be covering chapter 12 next week.